Protein synthesis, part 1, transcription. 1.
This is the nucleus. Protein synthesis starts with RNA synthesis in the nucleus. This is the DNA strand. In RNA synthesis, a strand of DNA is read. The DNA is in the shape of a double helix. This is the DNA strand unwound. This is the template strand. It is part of the DNA strand that is read. It gives a code to turn on certain genes to express certain traits after translation. And this is the TATA box, the promoter where transcription begins. This is the transcription initiation complex. The transcription initiation complex starts transcription. It pulls the DNA apart and joins together RNA nucleotides. It is made up of RNA polymerase 1 and 2 and transcription factors, which are helper proteins. The transcription initiation complex pulls apart the DNA strand and reads only the template strand. This strand is read from the 3' end to the 5' end. When the transcription initiation complex reads a stop codon, the messenger RNA strand is terminated. The RNA transcript is released and the transcription initiation complex detaches. In this mRNA strand, the UAA was the stop codon. The mRNA strand then carries a genetic message read off the DNA to ribosomes in the cytoplasm. A 5' cap and a poly A tail attach to the pre-mRNA strand and export it from the nucleus. These are exons and these are introns in the mRNA strand. The introns will be cut out and the exons will be read. RNA splicing removes the introns from the mRNA sequence and leaves the exons to be read in translation. There are small nuclear RNA and other proteins that go into the spliceosome that catalyze the excision on introns and joining of exons. The introns are cut out of the RNA sequence and the exons are spliced together by spliceosome.
Protein synthesis, part 2, translation.
Transfer RNA or tRNA transports the mRNA strand from the cytoplasm to ribosomes. The small ribosomal subunit attaches to the mRNA strand. The large ribosomal subunit then attaches to the strand. They begin to read the strand until they reach the start codon. The large ribosomal subunit has a P site which holds the tRNA that carries the growing polypeptide chain, an A site which holds the tRNA that carries the amino acid that will be added to the chain next, and an E site which is the exit site for each tRNA. After the start codon is read, the amino acyl synthetase sends a tRNA with anticodon UAC, which carries the amino acid methionine, to the P site, where the protein that is being coded will start being made. A process called hydrolysis is used to create GTP, an energy that makes the peptide bonding more accurate. An amino acyl synthetase is an enzyme that attaches the appropriate amino acid onto its tRNA. It works like a lock and key and will only attach the correct amino acid. While a codon is attached to the P site, the amino acyl synthetase sends the next tRNA to the A site to wait to be added to the polypeptide chain in the P site. The amino acid on the tRNA in the P site attaches to the amino acid on the tRNA in the A site. A hydrogen bond is formed between the two amino acids. The empty tRNA in the P site moves to the E site and is released. The tRNA in the A site moves to the P site and the mRNA strand moves along with it. The amino acyl synthetase then sends another tRNA with an amino acid attached to the A site. The same process is repeated. Until it reaches a stop codon. When a stop codon in the mRNA is reached, translation stops. A protein called release factor binds to the stop codon and the polypeptide is freed from the ribosome. And the tRNA enters the E site, then leaves the ribosomal subunits. Then the ribosomal subunits detach from the mRNA strand. Depending on the protein's code, it might have to be modified or sent somewhere before it is functioning.